Joining us today at our program, it's Big Data and Corn Genomics. What does that mean for you? And these sessions are brought to you by U of M Extension and generous support from the Minnesota Soybean Research and Promotion Council and the Minnesota Corn Research and Promotion Council. I'm Liz Stahl, I'm Extension Educator in Crops. I work here out of the Worthington Regional Office in southwestern Minnesota. And I want to welcome our speakers today. We have Dr. Candice Hirsch. She's our U of M agronomy professor and corn geneticist. And we also have Tom Hoverstad. He's a researcher at the Southern Research and Outreach Center in Waseca, and he'll be joining us during the discussion. I guess I will turn it over to Dr. Hirsch.
Well, I'm excited to talk about corn genomics today with all of you and what the use of big data in corn genomics and corn production looks like. So our group here at the University of Minnesota is the Maize Translational Genomics Group. And our big goal in our group is to utilize big data to understand the relationships between the genome, the environment, and the phenome or the plant yield or durability or whatever phenotype we're interested in, so that we can improve corn as a crop plant to grow under variable growing conditions. And so today we're going to mostly focus on thinking about big data in terms of genomic data and high throughput phenomic data. But there's a lot of other kinds of big data that get incorporated into thinking about how to improve corn for production. So a lot of times we sit and look at plants and we say, what made this plant look like this? In this particular environment, it has this phenotype at this yield. What made that happen? And if we can understand that, we can reverse engineer plants to be what we want them to be in different environments. And we can strategically put different hybrids into different growth environments where they'll best perform. And so when we are thinking about what makes this plant look like this or yield this, we first start with the DNA. And that's because this is really the blueprint of what makes the plant that we see in front of us. There's a lot of things that happen in terms of how that DNA interacts with the environment, but that DNA starts as the blueprint of what the plant is going to look like. And so in that light, this is kind of our first big data that we generate within our group, and this is sequencing data. And so what we do is we go out into the field, we sample plants, we isolate the DNA out of all of the cells and all of the sample that we collect, and then we send it to be sequenced. And what comes back is something that looks like this. And actually we get terabytes of data that look like this, where it's letter A, T, C's, and G's, and a bunch of them in the file. And we try to make sense out of what that means for the genotypic variation of that individual. And so here's an example of what this would look like on a single base between two individuals. So we have variety 1 and variety 2. And they're the same at many of the positions, but here's one where they're different. And so what we're doing is we're taking all of this data and we're looking at the 2.3 billion positions in the genome and comparing between any two varieties, how they differ. And eventually that helps us to understand the relationships between different individuals and allows us to predict their performance across different environments based on how different or similar they are, because the more similar they are, we would expect them to perform more similarly across different environments. So that's the genetic data that we generate. And in terms of how much variation there is in corn, so I think we're used to seeing fields like this, where everything looks pretty similar. And in fact, these are actually a bunch of different varieties in each of these different ranges and rows within this field. This is from one of our trial fields. This is our genetic nursery where things get a lot crazier and weirder between different varieties, the different lines that we're growing. But whether we're talking about this scenario or this scenario, the difference between any one of these two plants or these two plants is substantially different in the genome variation than between a human and a chimpanzee. So there's a ton of variation in corn that we need to be thinking about in terms of how we are breeding varieties for the future. And the variation that's here may not be everything that we need. We may need to be bringing in other types of varieties and different wild species that bring in different traits that will help to make hybrids for the future. So in our group, we think about not just the SNP variation or these single changes, we also think about what we call structural variation. And so what this is a part of the genome that's completely present or absent between two different individuals. So for example, we have a gene here, and in this genotype, we have the gene, and in this one, it's completely gone. Or we may have two copies. Here we have a single copy, and here we have two copies. And so we think a lot about this kind of variation as well. It's something that has really taken off a lot in the last couple of years as technology has changed and allowed this to actually be measured. And so just to kind of give you a feel for how much variation there is of this type, we looked across 26 different varieties of corn. In one variety, there's about 40,000 genes. If we look at two varieties and we say how many total unique genes are there, there's another maybe 15,000 that get added on. And if we look at three and so on, if we look at across these 26 genotypes or varieties, there's over 100,000 unique genes, whereas any given one only has 40. So that's 60,000 extra genes that are floating around, sometimes present, sometimes not within a genotype. And so this has major phenotypic consequences. I'm going to show my favorite example of this here. So this is the sugar enhancer story. So if you really like fresh market sweet corn and you really like that super sweet flavor of it, what we've shown is that there's the cause of that super sweet flavor is the deletion of a gene that if you have it or you don't have it makes you be super sweet or not super sweet. So that's just one example of it. There's a lot of examples of these structural variants that are important for aluminum, toxicity tolerance, or other types of adaptive and agronomic properties that are important. So big genomic data has revealed that there is a lot of variation in the genomes of different corn varieties, and that this variation is very important for phenotypic variation that consumers care about and that we care about in terms of production. And so all this variation is what's really led to yield increases over time. It's one of the main, a large contributing factor, definitely not the only. So as varieties have been selected over time for improvement, there's been changes to their genetic content, and this has driven yield increases along with other factors such as the environment. So agriculture doesn't exist in a vacuum by any stretch of the imagination, and plants, whether they're corn plants or any other plant, are sessile. They're sitting in whatever environment they germinated in, and that's what they have to adapt to. And so while we wish this was what every year looked like, perfect rain, perfect amount of temperature and GEDs and all of these things, the reality of growing corn in a production setting often looks more like this. And in the last couple of years, and definitely moving forward with climate change, we are going to see more and more of these not ideal environments. And so while we saw that kind of continuous yield increase over time, a few slides back, if you zoom in, there's these low years. And those low years are the results of droughts, floods, other climate events. And again, modeling has shown that there's predictions that these are going to increase in severity, and the projected yield changes are only going to increase over time. So that's where big data can also help us in understanding how different genotypes interact with the environment. And certainly not all genomes interact with the environment in the same way. So while we have that blueprint may be affected by different environmental factors more or less depending on what's in it. And so Minnesota is a prime example where we can see this every day with people, where it's freezing cold outside and some people are walking around in shorts and some people are totally fine to be, or freezing their butts off. And plants are the same way. You put two plants in one environment and you put them in another environment, and their yield or whatever trait we're measuring can change dramatically in terms of the ranks between those two individuals. And so we call that the genotype by environment interaction. And there's been a number of studies recently that have shown that different genes control a plant's potential versus its ability to react to its environment. So what is its blueprint saying is its potential, and then what do we actually observe across different environments? And other studies have shown that corn breeding may have reduced genome plasticity, so that ability to change based on the environment that an individual is placed into. And so while we used to measure plants at the end of the season with a stick, walking around the field, and we took a single end of season yield metric, and those were two of the biggest traits that corn breeders measured along with flowering time. We realized that we need to understand more about how plants interact with their environment to maximize both yield and plasticity. And so to do that, we need to measure traits at many times during the growing season and in different growing environments. Because phenology matters a lot. When that rain event happens matters a lot. And the subtlety of that is far more than maybe we originally thought as to how many GDDs into the season when a particular rain event happens, or a drought, or a high temperature, low temperature, et cetera. And so to address this, we use drones to be able to do very high resolution phenotyping. So here is a graduate student group, Dorothy Sweet, who is using a Phantom 4 RTK drone. And she flies this regularly throughout the growing season and is able to reconstruct fields throughout time. And so you can see the plants getting bigger and bigger throughout the season. And her and another graduate student, Sarah Dorado, developed an entire analysis pipeline where they do their flights, they take those images that come off the drone, and they reconstruct them into a 3D image of the field. And from that, they can figure out how tall every plant in the entire field is. And we can pull out the individual information for each plot. And so what I'm going to show you coming up here is that there's a lot of variation that's not captured by end of season traits that we capture with drone imagery. So for the study that I'm going to talk about, Dorothy looked at a panel of 500 different varieties that she grew over 4 different environments and flew them regularly. So here's an example of one individual plot where she's showing how the height has changed over the growing degree units in that particular season. And she did this for almost 4,000 plots and used that to look at how much variation there is in growth curves for individual varieties. And so what she saw, here's one example of a particular variety that shows very little variation from year to year. So we have each color is a different year and growth curve, and you can see that they are almost entirely the same in every season. Other varieties like this one have very different growth curves and get to very different points at the end of the season, versus this one has different growth curves throughout the season, but really gets to that same end of season height. And so what we get from this is that we might think that this one and this one have kind of equal stability or plasticity, but in reality, they're actually quite different when we look at them throughout the growing season. And so if we quantify how different these growth curves are, which is what she's showing here, in different combinations of years, so say 2018 versus 2019 in this first line, these genotypes that are in blue are very similar in their growth patterns, but these other ones that are in purple and into yellow show a lot more variation in their growth curves. And so we can start to actually quantify and map the genetic elements that are contributing to a variety being more stable or less stable in its growth patterns across environments. These curves were made off of plant height data, but there's other metrics that we can pull out of a drone image. And one of those is canopy cover. And what we see is that canopy cover is actually far more responsive to environmental changes than plant height. So this was work done by Julian Cooper. He took those same drone images, and instead of pulling out the heights of each plant, he figured out what is the canopy cover by masking out the soil and saying how much plant material is there in each image. And he fit curves again. And without going into the details of this complicated plot here, what I want to show you is that the times in development where year and year have an effect is quite different between these. So earlier in the season in plant height, a little later in canopy cover. And that G by E, so that interaction of a variety with its environment, is far greater, almost double the amount of variation explained by genotype by environment interactions for canopy cover as for plant height. So by using these different traits that we can pull out of drone imagery, we can understand different aspects of a plant's potential and its plasticity. So one question we wanted to ask is, can we actually use UAVs to study how plants interact with different environment and management conditions outside in a more structured way? And so we designed an experiment that used 12 different hybrids planted at 2 different planting dates. So, so that these ones would experience an environmental event, like a rain event, at a later developmental stage than these ones that were planted later. And then we planted them at different densities. We again flew drones over these different plots. And unfortunately, we had a massive wind event that knocked everything over. And so we said, well, let's see if we can use drone imagery to study, sorry, my lights are trying out, to study, there it is, to study lodging. And so that's exactly what we set out to do. And we did show that indeed that you can use drone imagery to study both lodging events and recovery very effectively. So essentially what we do is we fly so regularly that we have flights, within days of something like this happening in a field, where you can see here some plants manage to stay up. This is a variety that, these are 2-row plots. This one stayed up quite well. This one lodged, these ones lodged. And so we can fly, we have flights that happen before these events, we can fly right after they happen, and we can see all of these lodged plants in our reconstructed images. And we can look at the difference in the height from right before the event to right after to quantify how much lodging there actually was. And so instead of having to walk through the field and try to score this, which is very difficult when plants are all laying on the ground, we can do this via drone imagery. And so we can say how different was the height before and after this event. And you can see here in this particular experiment, the plants in the first planting date suffered far greater changes in their plant height, which is indicative of greater lodging. So they were a little older and they lodged more than these ones. We can also capture the recovery after a lodging event. So you know that when corn plants fall down, depending on what time in the season they are, if they are still able to, they can gooseneck back up after a lodging event. But when this happens, it really limits what kinds of phenotyping we can do moving forward, because we can no longer have rovers that go through the field. It's difficult to walk through these plots, but drones are great because they are aerial, and so it doesn't matter that there's a gooseneck plant here. We can still fly them. And so here we're looking at across those 2 different planting dates. We had them growing, This was that major lodging event. We can actually watch this particular genotype recover out of that lodging event. And we can do that for all of the different varieties that are grown in these experiments. And we can track not only how much they lodge, how quickly they recover, how much they're able to recover, and things like that. So going back to that original question, we tried to do this again in the following year, planted that same experiment, and actually did even more drone flights early in the season, and it lodged again. And so we just said, you know what, clearly somebody is telling us that we need to study lodging, and for good reason, because lodging can cause very significant yield losses. And so we took this as an opportunity to actually really look at what was underlying the variation that we were observing in lodging, and to kind of quantify why we cared so much about this. So we first looked at data from an experiment called the Genomes to Fields trials. This set of trials has over 180,000 field plots that have been grown in 162 unique environments with over 2,500 corn varieties. So that's a lot of data that we use to look at variation in lodging that occurs across different varieties. And so we can see as we go through the different years that some of these have very severe lodging events, others have less lodging events. But as we took that lodging data and compared it to the yield, we see in this set of experiments, about a 12 bushel per acre loss when we have partial to extreme lodging. And crop losses due to lodging are reported to range between 5 and 25%. So a substantial yield loss. And so something that we wanted to take advantage of since we had the data. So we asked the question, what factors influence lodging? And so again, in our experimental design, we have different factors that we could look at, from planting date to density to the varieties that were included in the study. And so we asked what factors were influencing lodging severity. And so here you can see our different planting densities, our different planting dates, the 2018 data, 2019 data, and the percent change in height. And so what we see is planting date 1 in 2018 had very severe lodging, planting date 2 in 2019 had very severe lodging. And if we overlay these colors, are overlaid by GDUs or GDDs, what we see is that these were at actually about the same number of GDUs accumulated within when these events happen. And so it's kind of a very vulnerable point in time for the plants to have a strong wind event. And so what we did was we modeled a bunch of different factors. So the genotype, the density, the planting date. We also looked at the pre-lodging plant height, the early season growth, the mid-season growth, and we looked at what factors were most important. And what we saw was that density was really important, as well as pre-lodging plant height, and then the early mid-season growth. And so this makes a lot of sense. With density, we have a very different root structure that results when we plant at very high densities. And that also forces different growth patterns to accommodate the high densities that the plants are being grown under. And so these were kind of all the factors that contributed to the severity of lodging that was observed. And the severity of lodging definitely came with a yield penalty. So this is looking at all of the different densities combined, and you can see that if we have moderate to severe lodging, that there is quite a yield penalty that comes with this. And if we look across the densities, this is most extreme at the high density, where then no lodging out to severe lodging comes with a more substantial yield penalty. So another quick study that I want to talk very briefly about is one that we looked at how drones may be able to be used in production fields. And this is one that's very early in very early stages of analysis, but just to touch briefly on it. So this is drone flights were conducted over production fields across 3 different years, 2020, 2021, and 2022. And the heights of plants throughout the field in each of these years, as well as the growth rates, were quantified off of those drone images. And what we observed when we compared both height and rate to yield, so these are correlations between each of these metrics and yield, we do see that there's times in the year that we can look at the height pretty early in development and have quite a high correlation to the yield of that particular part of the field. So here in 2020, like very, very early. But if we look across years, we can see in this part of development, we can use the height or the rate of growth to get pretty high correlations with the yield map for that particular field. And so the next thing we wanted to ask is not only can we correlate, but can we actually take that early data and predict what the yield will be very early on? This allows for management intervention early in the season that may combat some of these potential yield losses. And so what we see is if we make prediction models, for some years, we're able to predict very well. So here in 2020 and 2022, I don't know if I'd say very well, but we have some predictive capacity. In 2021, we had almost no predictive capacity. And without getting into a lot of the details on this for the sake of time today, what I'll say is that 2021 and the location that we were flying was in a severe drought situation. And far more of what was explaining the variation was the water table than anything else. And so because the other 2 environments were not in as severe of a drought situation, that wasn't really captured in the models that would be generated off of those years to predict it in 2021. And so what this tells us is that there's potential here, but we would need substantially more environments to be able to develop robust models that would allow us to predict end of season yield from very early development growth rates or plant heights. So kind of bringing this back to the beginning. So I said that we are really interested in using big data to understand the relationships between the genome, the environment, and phenotypic variation, with the goal of improving corn as a crop plant being grown under variable growth conditions. And I hope that you saw some different applications today of where we are using big data and where big data can be used not only in breeding context, but also in production contexts. Because ultimately, we care about trait variation for things that happen at the end of the season, the yield, the durability, things like that. But there's a lot of things that we can measure very early on, before a plant even goes in the ground, seed goes in the ground, to what it's doing throughout the season. That can give us a lot of information about how it's engaging with the environment and ultimately what it's going to look like at the end of the season. And so with that, I'm going to just acknowledge a number of different funding sources that have been very supportive of our program over the year. And I especially want to highlight the Minnesota Corn Growers Association and the National Corn Growers Association that have both been very supportive of our project. And here's just some fun photos of our group collecting all of this crazy data. And yes, this does sometimes happen where we are combining in the snow, just like everybody else. So with that, I'm happy to take questions.
Thank you, Candy. And one thing I forgot to mention when I was introducing you too, is I should mention you've been recently awarded with the University of Minnesota McKnight Presidential Professorship in Plant Genomics. So that is awesome. Again, we're very fortunate to have you at the University of Minnesota and very excited about all your work, even though I know even there's things that I don't totally understand everything either, but I know it's It's a good thing, you know, and it's interesting you brought up the corn lodging. I worked with a seed company, and this would have been like 25 years ago about now. We had a big wind event and we had all these hybrids out. And I remember, you know, looking at all the lodging out there too, and we were rating our plots. We didn't quite have tetrabytes of data. I mean, we had like did a scale of like 1 to 5 and collecting that information, trying to figure out it was very difficult. So things have really come a long way. That's what I'm trying to say is very interesting. But yeah, with all this, we did get us some questions about this too. And Tom, I'll bring you into this as well too, because I know you've been working with the plots as well. But one question was, what were the stages of corn for planting date 1 versus planting date 2 when the lodging occurred? So I suppose they're trying to see, was there a difference in the stage of the corn at that time?
Yeah, so I can't remember the exact vegetative stages that things were at anymore. I want to say it was like V5, V6 was the in the first season that didn't do so bad, and then V8 and 9 was when it did bad, and then again that was V8 and 9 was when it suffered a lot in that second year. So that was kind of the if I remember correctly, what those GDDs actually correlated to in vegetative stages. It was around that.
Okay. Yep, because I'm sure that'll make a difference. And then there's number of questions here about the UAVs. Just wondering, where are you flying them and how many flights are you doing in a season when you're trying to determine a yield correlation? I'll kind of combine those two questions, I think.
Yeah, so different studies are flown in different locations, but for the stuff that I was talking about, that was in Waseca, and we were flying at least once a week over the production field. Up until flowering time. And then other work that we're doing as part of the Genomes to Fields collaboration actually has drone flights on plots that are growing from Texas all the way up to Guelph, Canada, and Nebraska, all the way east. And so, and different genotypes that are adapted to those different environments, I should say. It's not the same thing growing everywhere. But we have drone data flown at least weekly at all of those locations that, as well as combine drone flights weekly, combine data at the end of the season. And that's what we're working on right now to try to, once we realize how many more environments we needed, we move to that data set. And that's super early right now, and we don't have much to say.
Okay, interesting. And there's a question, just a point of clarification. The corn plant has 40,000 genes and there are an additional 60,000 genes available for a total of 100,000 in the corn gene cloud. Is that about correct?
Yeah, yes. So if we took one plant and we looked at its genome and we said, well, how many genes are in it? There'd be about 40,000. But if I took all of the genomes and I said, what are all the genes in them? And then I made a unique non-redundant set. So if one gene was found in multiple individuals, I only counted it once, then there's 100,000. So if I look that basically what I want you to take away is that there's a ton of genetic variation that the gene doesn't even exist in an individual, let alone differences in the sequence of that particular gene.
Interesting. Thank you. And oops. So, and Tom, I'm going to bring you in here too before we hit some more of these questions as well too, because I know you've worked with a lot of the plots. Of course, you're a researcher again at the Southern Research and Outreach Center in Waseca. Been there many years. Have a lot of great information to share as well. Just wondering if you want to share a little bit how, like I know you work with the corn variety trials too. So that's kind of taking all this information and then getting it out to the field and something that farmers can get their hands on. If you just want to share a little bit about that, how that works and how this all ties together. And
Sure, we've had the opportunity to provide a location for Candy to do a lot of her research, and a couple of things I'm reminded of. Drone imagery sounds like it's and she's right. Walking through fields in that lodge corn to get scores for lodging is difficult. You can't use a robot to do it. Drones work good for that. But remember, drones can't fly every day. There's some days when their drone crew came down and all of a sudden the wind's blowing 30 miles an hour, and you have to take that in consideration too. So we've had the opportunity to work with Candy, and I really enjoy that. She's got a big trial that we always do, and you really get to see a lot of unique corn. Like she said, there's corn that's probably more adapted to maybe Missouri than here, and there's also some that maybe that's adapted here. When you grow it in Missouri, it looks entirely different. So they're getting a lot of information on that, and it's really fun to be a location to help with that. We also, on the little more of the applied side, probably what you'd call small data, is we do a corn performance test across the state. And those, I think Liz could put a link to the results from that in the chat box. I think you can find that. We go as far north as Crookston, and we have a couple sites up north. We have central Minnesota, southern Minnesota, and I also get to see the genetic variation in those. A lot of times we plant some of the 85-degree or 85-day corn that is adapted for Staples or Crookston, put it in the borders here, and it looks entirely different. So where you're growing corn makes a lot of difference in where it's grown and tested and adapted. I take those 85-day corns to Staples, and they look terrific. Look at them here, and you'd say, I'm not growing that corn. So we get to see that also. And I think her data is studying that in much more of a quantitative method than just looking at it.
All right, great, thank you. And yes, we'll get that link in the chat, and also there'll be a Google link at the very end, and we'll have kind of all these resources together too. Diane, if you can help me out with getting those links in there, because I can't talk and chew gum at the same time here. But anyway, and some other questions too. Well, there's a question about like short corn hybrids, and I'll open this up to both of you too. Have you looked at like the short stature? I know there are some people at the university that are looking, like Rex Bernardo has been working with that. Are you looking at that as well? And seeing if that does have anything to do with lodging, and how does that play with lodging and yield prediction models?
We did a short corn trial with Jeff Coulter. Some of the material from the Bayer side of it, which came just from traditional breeding. They've got some that grow shorter, and they looked at that. I think Candice could probably address more of the ones that involve gene editing or gene transfer and those kind of things. But we did a trial, and the attractive feature would be one, it's going to lodge less. I think you could just gather that from information that Candy had on some of her corn, that the shorter it is, the less likely it's going to be hit by wind. It also makes it convenient for some of our mid to late season applications. You can get over it with ground equipment a lot easier. We looked at it this year. I think this season, probably it was planted later, had to clear a lot of hurdles with how we can use that based on proprietary information. So we didn't plant it till later, and I think it yielded about the same as conventional corn, but I think the yield potential probably wasn't realized because we didn't plant it at the best time of year. It did fine. It grows shorter, no question about it. 30 inches shorter. We measured that the old-fashioned way. But one thing I did notice is it seems the leaf canopy in there is very dense. I think that might impact some of our management practices. It would be a great environment for leaf diseases when the leaves are packed in there as tight as they are. So I think it's on the early side. There'll be more information on that, but there's certainly potential there for those that oftentimes get high wind events. That might be an attractive genetic feature for those growers.
Yeah, and I'll just add, I mean, most of the data that's available is directly from the companies because it hasn't been, this is the first season that people have really been able to test it that aren't at the company. So most of the data that we're going off of is from the people who are selling it, which is where Tom's work is so important to really test these by an unbiased party. But some of what Tom was talking about is important to note is that reduced height doesn't come because there's less nodes and therefore less leaves. It comes because the inner nodes are shorter. And so that does have its different complications, but most of their data is showing comparable or improved yield, and reduced lodging is the data that's coming from companies.
Great. And one question we did have was, do you know if corn rootworm was a factor in the lodging as well? Was that playing a role in there?
In our study, no. But certainly can be a major factor in depending on the resistance of the varieties that are grown.
All right, great. And here's a question too. Any theories on why artificial selection and modification seems to interrupt genome plasticity?
I think you get what you breed for. And so if you're very focused on breeding for one thing, and that is increasing yield across your test environments and like the mean yield, and you're doing that in a time when there was not as much environmental variability as there is now, you get what you select for. Breeding is really effective at getting what you ask it to give you. And if you didn't ask for plasticity, you're not going to get that.
Yeah, that's interesting. And that brings up, we had a number of questions that came in at registration as well too that relate to this topic, this plasticity. Any projections what climate change is going to mean for production in Minnesota? I mean, we know the climate's been changing. Of course, we had a couple drought years. We had some wet years before that. But again, you look at the long-term trends. It's great to hear how much plasticity we have in the corn genome, but any projections what this is going to mean?
Oh, I think I can address that a little bit. I've had the opportunity to work with corn breeding and a number of breeders throughout the years, and I'm often reminded that, sure, it's hard to deny climate change. The climate is changing, but also corn genetics change just as fast. And corn is bred, selected, and tested in the environment it's going to be grown in. So as climate changes, I think corn genetics will change because they really only last a couple, 3 years. And now we're moving on. Yields are going up 2.5 bushel per acre per year. And people are growing the new hybrids that are selected in that environment. In addition to that, I mean, if the climate changes drastically, farmers will change. I think if you compare what farms looked like 50 years ago to what they look like now, farmers have adapted to a changing economic climate. So bottom line, I feel is, sure, the climate's changing, but this area will still be an important agricultural production area. What it looks like might be different than what it looks like today, but it'll still be agricultural. Unless somebody builds an airport on your farm, it's still going to be a farm.
That's right.
I completely agree with that. I just want to add one thing that makes it challenging is when the environment changes at too fast of a rate, because the crosses that are made to the initial cross that will eventually generate a hybrid, there's years and years between that, from that initial cross to making selections, to doing testing, to increasing seed. It's a many years. And so the challenge that breeders are going to have coming up is decreasing the time for the breeding process so that it can happen as fast as climate change is happening.
That's a great question too. I mean, how many years, again, do you figure it takes? I mean, I think that's probably changed over time now when you have more tools, but how many years do you think it takes to have, from a cross to actually having a hybrid out there that somebody can use on the farm?
I think Candice is
Yes.
better to answer
Yeah.
that. She's a real breeder.
I remember the guess, wasn't
I
it
just
like
get it
8
when
years
it's
or something
ready.
before? And
I get it when it's ready for testing. I'm not involved in the crosses and all that, but Candice would have a good feel for that.
Yeah, I mean, in some cases, crosses are made 8 to 10 years before a variety is actually the initial cross that will make that variety. As you go through all of the, you have to make crosses, you have to, do early testing, late, I mean, there's a lot. So it can be quite a long process, which is where the rapid change in the climate that we are experiencing now is harder to keep up with because the crosses are not happening now that are going to be the varieties for next year.
We got to be able to predict the future, right? As best you can. And how about did have a question come in earlier too about is there an updated corn maturity rating map for Minnesota? Just wondering, has that changed? And is that enough for growers to reconsider maturity rating choices?
Well, that's a good question. If you just think about it on one level, our climate is getting warmer and it's getting wetter. And if you'd think because we're getting probably more growing degree days, that longer maturity hybrids would be more common, but I really don't see it. A couple of reasons I think is our risk of frost really hasn't changed. I mean, and also when you look at the warmer conditions we're experiencing, it's more dramatic in the winter than in the summer. So I don't think our summers are that much warmer. I don't think, and if we are warmer in the spring, it's usually conditions that allow us to plant more so than temperature. We usually have to wait for soils to dry out. We need to have the right conditions to plant. So having a few more growing degree units in the spring of the year probably doesn't really impact how we plant corn. If we get a few more growing degree units later in the season, I think the solar radiation we're getting isn't quite the same as the summer. So I think we really haven't moved our maturity rating and what's adapted that much based on climate change. I think the maturity recommendations are still quite the same. The thing we have enjoyed mostly the last few seasons with extra growing degree units is drier corn in the fall, less we need to dry. So I don't see it climate change really changing our relative maturity scale much at all. And the risk of frost in the fall is still there. So even though we may see warmer conditions, I don't think our frost dates have changed that much. We still have the variation that we're used to here in Minnesota, and it can freeze in the middle of September on any given year.
True, good points. And here's a question kind of, and maybe you've addressed this a little bit already too, Candy, but again, a question that came in earlier. How can we bring together big data to solve crop production problems across industries?
Well, across industries is challenging. But I would say in general, the use of big data is allowing us to maximize yield increases. So as Tom said, we're still getting consistent yield increases, but that gets harder as the plant gets pushed more and more to its maximum potential. And so to keep making those improvements, we really have to start expanding our toolbox beyond measuring yield at the end of the season, because that's, that gets you a lot of progress when there's a lot of progress to be made. But as the plant is more challenged to produce more and more with less inputs and more stressful conditions, we need to maximize what we know about it. Selection also works on the numbers game. So the more individuals you can test, the better chance that you're going to find the best individuals. So big data is really important in that way, because we don't actually need to test every individual in every environment to get an estimate of what its yield is going to be in that environment. And so we can consider a very, very large number of progeny for selection without actually having to grow all of them as a product of big data in agriculture, primarily through genomic prediction and selection.
Yeah, and you remind me of a question I wanted to ask you earlier too, because we know it's not, even though sometimes it seems all about yield, it's not totally all about yield, right? There's some other traits that you are selecting for, correct? And like looking at grain durability and things like that. Would you want to just take a little bit just to share kind of some of that in a nutshell?
You know, with 30 minutes, I had to like really
yeah,
pick and choose. But yes, we're doing a lot of work thinking about the composition of the grain. And this is for different reasons that we are thinking about it. One aspect is on food grade corn. So we work a lot with companies like PepsiCo that make corn-based food products for direct human consumption. So things like Doritos, and how the composition of the grain impacts the cooking process and ultimately the product that the consumer is eating. This has impacts on human health, how much oil is taken up, carcinogens that get formed along the way. There's a lot of things that we think about in that space. We also think about composition and different aspects of the grain for durability, as you mentioned. And that's work funded by Minnesota Corn Growers and National Corn Growers Association to try to understand what has changed over time that has reduced the durability of grain. And this has major economic impacts on grades that corn gets as it's going through various markets. And so we are looking at not just the composition. We know that protein has decreased and starch has increased. That's a great way to increase yield, is to have a lower energy molecule in the grain, and it's a lot less energy to make starch than it is to make protein. So we know that that's changed, but we're looking at other aspects of the grain as well. Has the pericarp thinned? Is the shape of the kernel, how fast things are having to dry down in the field, how wet stuff is coming off out of the field, all these different factors that can contribute to pericarp cracking that results in a more vulnerable kernel as it's moving by boat and rail and in and out of shipping containers and these sorts of things. And so we have a big project right now that many of the growers around the state have contributed to, and we are so appreciative, where they have sent samples that they've grown on their farm, and sent a lot of metadata. So what variety they grew, when they planted it, when they harvested, what management practices they used, so that we can conduct a big multi-environment study around the state to look at factors that may be contributing to better or worse durability.
Is that something you'll be doing this fall again too? Do you predict, or unsure yet, or?
TBD.
Okay, well, stay tuned. We'll know
Yeah.
more about that later. Great. And I do have another question here too that came in. What do you think contributed to the great corn yields of 2023? I mean, here we had a drought over all these areas. Is this part of all the big plasticity that we have in the genome here?
Oh, I think the breeders would like to stand up and take all the credit for that, but I also think Mother Nature was responsible. Now, you mentioned great corn yields in 2023, and some people did get very good corn yields when the news was all about how dry it was, drought taking over the state. But I do want to remind people of a couple things. We didn't really get below our annual precipitation until about the first week of June. Right here, I'm in Waseca, south central Minnesota. We had a very good soil recharge. We have native soils that hold a lot of water. We had a lot of rain in the spring of the year, more than normal in May. Didn't really turn dry till later. So we had a lot of moisture in the soil, and we're blessed with soils that hold a lot of water. Some year that's a curse and not a bless. We have too much water. But this year, I think our water holding capacity helped out. And another thing, we had 2.5 degrees cooler than normal in July, which I think helped out our corn crop. Now, having said that, there was some areas of the state where they're saying we didn't get that great crop. Down in the southeast part, probably centered maybe from Rochester down to Iowa. I think they missed out on some rains. We had decent rain in August that I think they missed. They maybe didn't get quite the recharge we had, and maybe they don't hold quite as much as some of our soils. There's areas that did suffer from the drought. National or state average corn yields were down about 10 bushel per acre from the previous year. So it wasn't everywhere, but we were fortunate to get what we had. And I think Mother Nature does explain some of that. Not to minimize what the breeders are doing, but Mother Nature still has the final say. They're working on how we can how we can do the best with what Mother Nature gives us. And we certainly appreciate that. I like working in Candice plots, thinking that someday a grower is going to have 300 bushel corn, and this was all part of it. How we learned what is important to do that. And I tell our students that too. I tell them, well, remember you were here when you can go home and tell your father. A lot of our kids are farm kids, and I say, well, tell your father that you worked on corn, and when he's growing 300 bushel corn, you can say you helped him with that.
That's right. And that is one of our questions, how to grow 300 bushel corn. So I think, was that one of the, did we have some information? Did Jeff Coulter, our corn agronomist, work with some plots with that?
Well, he did a
And
trial, and I called it the high yield trial. He got some funding from the corn growers and looked at adding extra inputs. We did starter fertilizer. We side dressed with extra nitrogen. My take from that whole trial was when you get high yields, it's because of what the season gave you, more so than just adding inputs beyond what you need. We saw some really good yields over in the Rochester area from some of the lower input treatments, and that soil provided a lot that year. So my bottom line is stay with it. You'll grow 300 bushel corn. We're going up 2.5 bushel per acre per year. You might have to stay with it 40 years or so. I'm 64 years old. I might not see it, but somebody's going to. 20 to 40 years, I think 300 bushel corn on the farm will be common.
well, we're getting near the end of our time. Are there any last comments you want to add here, Candy, before we wrap it up? And then I'll ask you the same thing, Tom, after that. Um, but any parting comments here?
Nope,
No,
I
just
think
thank you for inviting me to present. This was an honor and excited to share the work we're doing.
Yep, doing a lot of exciting work. Like you said, our kids someday will be like, hey, I know how this all got going here. But
yep,
and Tom
we'll
is yeah. Oh,
we'll
go
have
ahead.
300 bushel corn, and it's probably going to be because of genetic gain. So that's where it is.
And do you have any recommendations before we wrap this up too about like selection tools, hybrid selection tools for farmers in Minnesota?
Oh, sure. Of course, I believe in our trials. We look at a lot of locations and try to find trials that are randomized and replicated. Those are principles that really address getting at genetic potential instead of environmental differences. They aren't always easy to find. Our trials are that way. There are some farmer initiated trials. The first trials, I think, use those basic statistical principles that are necessary. I think that's a good source of information. The corn growers usually, if you go to the Minnesota Corn Growers website, they have a number of locations. You can kind of use locations as replications for them. They have a good database. I also believe that talking to your seed dealers is a good idea. They want you as a customer. They want you to have the best information you can get, and also your own experience. Now, don't stick with a hybrid too long because corn yields are going up. Don't get emotionally attached to a single hybrid, because if you do that for 3 years, you're going to be about 7.5 bushels behind. One thing I would like to encourage people when they do look at hybrid data, just do one little thing. Most people kind of look at it to see what the highest yielding hybrids are. Do this little bit of a turn and say, I want to use this data to predict what's going to be the highest yielding hybrid next year. And that often doesn't include the trial that's right next to my farm. Look at as many locations as you can, and I think that's going to be a better indicator of what's going to be best next year. This year is history. It isn't going to happen again.
Isn't that right? And yes, I had a comment here, and I'm sorry, I was going to add this later too. Yes, Southwestern Minnesota, we also had the question came in obviously from somebody about the yield. They didn't get hit as bad, people in southwestern Minnesota also had significant yield losses. Again, it depended when you get the rain, how much rain you get, and some areas just didn't time out well at all. So yes, we were hit in other areas of the state too. So yes, thank you for pointing that out. I wanted to mention that. I didn't do that. But anyway, again, well, I'm going to wrap things up here. Again, thanks everybody for attending today. Thank you to our speakers today. Much appreciate your great information.